Hello everyone, welcome. So today I will be discussing about biotechnology, principles and processes, part 3 cloning vectors and multiple cloning sites. I've already discussed about what is biotechnology, what are the various principles of biotechnology, the tools of RNA technology, which consist of the restriction enzyme, the cloning vector, multiple cloning site, host cell, everything. So now going ahead, we will be coming ahead with the third part, which is cloning vectors and multiple cloning sites. Coming to cloning vectors, the molecular analysis of DNA has been made possible by cloning of DNA. The two molecules that are required for cloning are DNA, which is to be cloned, and the cloning vector. So I need the DNA, which is my which is my desired or gene of interest, and the cloning vector, which is my target. Okay, a cloning vector is a small piece of DNA that which is taken from a virus, or it can be a plasmid or a cell of higher organism that can be stably maintained in an organism, and into which a foreign DNA fragment can be inserted for cloning purpose. Most vectors, they are genetically engineered, and cloning vector is chosen according to the size and the type of DNA to be cloned. So what are the various characteristics of a cloning vector we'll have? For a cloning vector, I need to have the origin of replication, which is ORI. So I need ORI, the origin of replication, which is specific sequence of nucleotide from where the replication starts. It should have a selective marker. I have selective markers which are there for me to select the recombinants. Then there should be restriction sites. So there are numerous restriction sites. I have a synthetic multiple cloning site can be inserted into a vector, replicate inside the host cell to form multiple copies of recombinant DNA, and it should have less than 10 kb in size. Now coming to ORI, what is origin of replication? Origin of replication is a sequence from where the replication is going to start. And any piece of DNA, when linked to this sequence, can be made to replicate within the host cell. So this sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose ORI or origin will support high copy number. So it is not only to initiate the replication, but also ORI is responsible to maintain the copy number. Next, I told you there are selective markers. So if I talk about selective markers, this selective marker is required for maintenance of the plasmid in the cells. Because of the presence of the selective marker, the plasmid becomes useful for the cell. Now when I talk about selective marker, so in addition to ORI, the vector, it requires selective marker which helps in identifying and eliminating non-transformants. When I say transformants, means those which are having my gene of interest in the plasmid. Those which are not having is the non-transformants, and selectively permitting the growth of transformants. So I can always differentiate the transformants from the non-transformants. Transformation is the procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding the resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, they are useful as selective markers for E. coli. So normal E. coli do not carry resistance against any of these antibiotics. Alright, now Another thing we were talking about, we have talked about ORI. We have talked about selective marker. The third one is what? Multiple cloning site. So if I talk about multiple cloning site, many cloning vectors contain multiple cloning site or polylinkers. So a DNA segment with several unique size restriction endonuclease located next to each other. So restriction sites of polylinkers are not present anywhere else in plasmid. Cutting the plasmid with any one of these restriction enzyme that recognize a site in the polylinker does not disrupt any of the essential features of the vector. Now coming to one plasmid, which is PBR322. P stands for plasmid, B and R are for the scientist, the name of the scientist, which is Francisco Bolivar Zapata, which is Bolivar for B. Rodriguez is for R. Raymond L. Rodriguez. They were the scientists who designed this PBR 322, and 322 is the order of the synthesis that distinguishes it from other plasmids synthesized in the same laboratory. So this is how I name PBR 322 or any plasmids. If you see here, I have got the ORI. Then after that, what I have got the ampicillin resistance gene that I have. Ampicillin resistance is containing two restriction endonuclease, which is PVU1 and PST1. I have got a multiple cloning site. This is my multiple cloning site, which has EcoR1, CLAV1 and HIN3. Tetracycline resistance has got BAMH1 and SAL1 restriction enzyme, and I have a ROP that has PVU2. So here in PBR322, I can see I have got an ORI, which is origin of replication. For me to start the replication, I require ORI. Then I have got selective markers, ampicillin resistance, that is antibiotic resistance. I have ampicillin as well as tetracycline resistance, which is got restriction sites of PVU1, PST1 and BM1, SAL1 respectively. And I have a ROP area. ROP is regulator, operator and promoter, which has got the restriction site of PVU2. So when I was talking about antibiotic resistance, I was telling you I should be able to select the transformed cells. So if in a master plate I would have cells which have not been transformed, like these cells did not take up the foreign DNA. Okay, there might be cells which are transformed. Like if I talk about the transformed cell, this transformed cell, they may be transformed with non-recombinant, so they are not, recombination did not take place, and there would be ones where recombination would have taken place. So when I talk about these kind of transformants, then what do I say? See, non-transformed cells means I can always understand there is no transformation. It did not, it is the plasmid as it is. There is no change that has occurred. No restriction has occurred. Okay, the restriction. So I was talking about ROP, right? ROP. So, so they have a lag Z or lag Z, so which is going to produce beta galactosidase. So this beta galactosidase, if you E. coli strains, they are deficient in beta galactosidase function. So and a vector that is competent in beta galactosidase function, the cells are grown on X-gal. Once they are grown on X-gal, which is a dye-linked substrate, when cleaved from X-gal, the blue pigment is insoluble. So there is a blue pigment which is produced. So I have a cloning site. In the cloning site, I introduce my beta galactosidase gene, and my foreign DNA is there. So my beta galactosidase gene is there. In beta galactosidase gene or beta gal gene or LAGZ, I have got a cloning site. I'm going to restrict it. I'm going to add my foreign DNA. So there are three chances that can occur. My insert is within the LAGZ. My insert can be outside LAGZ, and there might be no insert, that is non-recombinant. So if I have non-transformed cells, then what will happen? My beta gal gene will be very much active. So it will produce blue colonies because it is dilinked. So it will produce blue colonies. If the gene of interest is outside, again when I say it is recombinant, not with my gene of interest, then what will happen? Again my beta gal gene is not being disrupted, it will produce blue colonies. Only and only my recombinants with the gene of interest in the beta gal will only produce white colonies. So I can select off the white colonies and I can go ahead proceeding next. So the next we will be going ahead uh, with the transformed cells, how we are going to divide it, how we are going to clone it. So that is the next topic we'll be going ahead. Till then, thank you.